Oxidative phosphorylation involves chemiosmosis and the electron transport chain. The ETC is embedded within the inner mitochondrial membrane. It establishes a hydrogen ion gradient, which is used to power ATP synthesis. Chemiosmosis will use the energy stored in the hydrogen ion gradient to drive ATP synthase, which will result in 28 ATP.
The ETC establishes a hydrogen ion gradient from low to high. The purpose of the electron transport chain is to replenish our NADH and our FADH2. It flows through the intermitochondrial matrix, then the FADH and the FADH2 are then replenished back to their NADH plus and their FAD. As our electrons continue to move through the ETC, an oxygen atom accepts 2 electrons from the chain and picks up 2 hydrogen atoms from surrounding solutions, forming H2O. The hydrogen ions from the intermembrane space flow from high to low through the ATP synthase, powering the synthase to phosphorylate ADP plus a phosphate group into ATP.
Fermentation provides an anaerobic path for recycling NADH back to NAD+, so that the cell can continue to make more ATP when oxygen is low or insufficient. There are 2 types of fermentation, lactic acid and alcohol. Lactic acid fermentation is when your muscle cells and certain bacteria can generate NAD+ by this process. NADH is oxidized back to NAD+ as pyruvate is reduced to 2 3-carbon molecules of lactate. Alcohol fermentation is when yeasts and certain bacteria recycle their NADH back to NAD+, while converting pyruvate to CO2 and 2 2-carbon molecules of ethanol. Here we have 2 side-by-side processes of fermentation. On the left, you can see that we will end with 2-lactate. We start out with a 6-carbon molecule of glucose. That glucose molecule goes through glycolysis, which we've discussed earlier, creating 2 pyruvate molecules. There you see that the NADH is oxidized to NAD+. It then forms 2 3-carbon molecules of lactate. On the right, you see that we also start out with a 6-carbon molecule of glucose. It as well goes through glycolysis, creating 2 3-carbon pyruvate molecules. However, in ethanol, CO2 is released, 2 molecules of CO2, which formulates 2 2-carbon molecules of ethanol. Gluconeogenesis means creating new sugars. The role is to provide glucose for export to tissues when glycogen stores are gone and there are no dietary sugar available. The precursors are lactate, pyruvate, glycerol, and amino acids. This process takes place in the muscles.
Using the glycogen stored in the liver and the muscles, the glucose monomer is phosphorylated as it is removed, producing glucose 1-phosphate, which is then converted to glucose 6-phosphate, as we saw in glycolysis earlier. This is the backwards process of glycolysis. Instead of breaking sugars, we're making the new sugars.
Photosynthesis is the process by which plants, algae, and some protists and prokaryotes convert light energy to chemical energy that is stored in sugars made from carbon dioxide and water. The stages of photosynthesis are when light and water enter the cell in the thylakoid and release O2 with help of the Calvin cycle, which takes in the O2 and cycles it into sugar. These 2 processes circulate ADP and NADP from the Calvin cycle into ATP and NADPH. And the cycle continues. One cannot work without the other. This is also known as coupling. The products released from photosynthesis are ATP, NADPH, and O2. Photosynthesis takes place in the chloroplast. The light reaction part takes place in the thylakoid membrane, whereas the Calvin cycle takes place in the stoma.
A photon excites an electron within a pigment molecule of the photosystem II. Those electrons are donated to the primary electron acceptor within the photosystem II, leaving electron holes, also known as charge separation. The electron holes in the photosystem II are filled the electrons from H2O, producing O2. The electron flows down the ETC to photosystem I. The electron transport carrier creates a hydrogen ion gradient that powers ATP synthesis. Hydrogen ions are actively pumped from the stroma from a low hydrogen gradient to the thylakoid space, which is a high hydrogen ion gradient. Hydrogen ions diffuse from the thylakoid space to the stroma through ATP synthase, producing ATP, also known as chemiosmosis. A photon excites the electron within a pigment molecule of the photosystem I. Captures of a proton by a pigment molecule. These electrons are then donated to the primary electron acceptor of the photosystem I and then to NADPH, of photosystem I and then to reducing it to NADPH. The electron holes of the photosystem I are filled by electrons from the photosystem II. The light reaction products are ATP, the NADPH, and O2. The ATP is then used to drive the Calvin cycle, which produces sugar. The input of the Calvin cycle is 3 CO2s, 9 ATP, and 6 NADPH, just to make one G3P molecule. The stages of the Calvin cycle turn 6 times. It starts with the carbon fixation, the incorporation of carbon from CO2 into organic compounds. Second is the reduction. 3 is the release of one G3P molecule. And fourth is the regeneration of the starting material. This ultimately makes 1 G3P, 9 ADP, and 6 NADP+.